In nutritional biochemistry and longevity science, the grapevine, Vitis vinifera, holds a revered status due to its extraordinary concentration of bioactive polyphenols. The primary compound driving this longevity paradigm is trans-resveratrol, a natural phytoalexin concentrated heavily within the skins of dark red and purple grapes. At the cellular level, resveratrol acts as a potent allosteric activator of sirtuin-1, a master NAD-dependent deacetylase enzyme. Activation of SIRT1 triggers downstream deacetylation of PGC1-alpha, initiating mitochondrial biogenesis, the creation of fresh, highly efficient cellular powerhouses. Concurrently, grape polyphenols upregulate autophagy, the natural cellular housekeeping process that clears damaged proteins and senescent cell debris. By mitigating oxidative DNA damage and enhancing telomere stability, dietary grape compounds deliver profound cellular rejuvenation from the deep microscopic level outward. The cardiovascular benefits of grapes represent one of the most thoroughly validated arenas of modern functional cardiology. The unique synergy of anthocyanins, quercetin, catechin, and oligomeric proanthocyanidins directly stimulates endothelial nitric oxide synthase. This enzymatic pathway amplifies the endogenous synthesis of nitric oxide gas, signaling vascular smooth muscle cells to relax and dilate. As vascular resistance decreases, systemic blood pressure normalizes, protecting delicate microvessels against shear stress. Furthermore, grape polyphenols exert a powerful shielding effect against the oxidation of low-density lipoprotein, LDL, cholesterol, the critical initiating event in atherosclerotic plaque formation. Paired with mild antiplatelet aggregating properties, regular consumption of whole grapes preserves flexible, elastic arterial compliance and substantially optimizes long-term myocardial perfusion. In ophthalmology, the human retina possesses one of the highest metabolic turnover rates of any tissue in the body, making it uniquely vulnerable to photo-oxidative stress. Grapes deliver an essential combination of dietary carotenoids, including lutein and zeaxanthin, paired with specialized flavonoid antioxidants that selectively concentrate in the macula lutea. These compounds act as internal optical filters, absorbing harmful high-energy blue light and neutralizing singlet oxygen-free radicals generated by daily digital screen exposure and solar radiation. Clinical trials indicate that grape consumption enhances ocular microvascular circulation, nourishing the photoreceptor cells and sustaining retinal pigment epithelial integrity. This dual bioshield plays a vital preventative role against age-related macular degeneration, diabetic retinopathy, and premature visual fatigue. The neuroprotective potential of whole grapes centers on the remarkable ability of low molecular weight grape polyphenols to cross the blood-brain barrier. Once within neural tissue, these molecules selectively concentrate in the hippocampus and cerebral cortex, the primary anatomical regions governing memory encoding, recall, and executive function. Grape bioactives stimulate the expression of brain-derived neurotrophic factor, BDNF, a vital protein that drives neurogenesis and synaptic plasticity. Concurrently, resveratrol and anthocyanins suppress microglial overactivation, mitigating chronic low-grade neuroinflammation and inhibiting the pathological aggregation of beta-amyloid proteins. Regular dietary inclusion correlates with improved verbal memory performance, faster spatial recall, and heightened cognitive resilience against age-related neurodegenerative decline. To harness the complete clinical potency of grapes, strategic varietal selection is key. Deep red and midnight black grapes contain up to 12 times higher polyphenol and anthocyanin densities compared to green table grapes, making them the superior choice for cardiovascular and longevity goals. If consuming seeded varieties, lightly chewing the seeds releases oligomeric proanthocyanidins, OPCs, some of the most potent natural free radical scavengers known to science. The evidence-based Master Daily Protocol recommends 1 cup, approximately 150 grams, of fresh, thoroughly washed dark grapes daily. For individuals monitoring blood glucose, pair grapes with a handful of raw walnuts or pumpkin seeds to decelerate gastric emptying and flatten glycemic excursions. Integrate this ancient fruit consistently into your daily lifestyle to fortify cellular defenses, nurture your microvasculature, and revitalize metabolic vitality. Medical disclaimer. The information presented in this documentary is strictly for educational and informational purposes and does not constitute medical advice. Always consult a qualified physician or healthcare provider before making any changes to your diet, medication, or medical treatment plan.